All right, we're going to start part 2 of nervous system. Here we go. So we're on slide 24, and we're just going to keep on going. Here we go. So we revisited the last thing we did was revisit the action potentials. Okay, so that's the first box. And the second box is graded potentials. Where do they occur? So I'll probably ask that in the chat in class. Where do they occur? What part of the neuron do they occur on? What purpose do they serve? What's their function? And how do they relate to action potentials? Those are all good questions for us to answer in the chat. Okay. Okay. Okay, so we're going to start with channels and transporters. It's nice to go over, even though we've started going over them, it's nice to go over them. So we have the sodium-potassium exchange pump. Remember, that's part of what we used at resting potential. Sodium-potassium exchange pump, it does require ATP. It pumps 3 sodium out and 2 potassium in. And it is part of our resting potential. Leak channels, they're open, they're closed, they let stuff in and out. And so we have potassium leak channels and sodium leak channels. Very nice. We have gated channels. So whenever you see anything that says ligand gated channels, a ligand is just a molecule. So a ligand gated channel would be an insulin channel is a ligand gated channel. A acetylcholine channel. So at the bottom of at the back end of that synaptic cleft, okay, at a neuromuscular junction, you have ligand gated channels. What's the ligand that they're grabbing? Acetylcholine. Okay, so those little acetylcholine on the motor end plate, on the skeletal muscle fiber, those are ligand gated channels. Okay. You also have these things called mechanically gated channels that you actually do know about, but we won't talk much about it until 232. But mechanically gated channels, when they get distorted, when the actual cell changes shape, they send a signal. Okay, so now, and you'll learn more about this in 232, but I will just say that tactile discs and tactile corpuscles and lamellar corpuscles, they are all mechanically gated. So you'll worry about it later. Okay, recall the all or nothing principle. Okay, recall the all or nothing principle. Write it down in your own words. What does it mean to have I always think of the toilet flushing, but what is the all or nothing principle and what is it relating to? Okay. All right, and then on this, what's so cool about this slide here is that it's showing something called the refractory period. So there's something called the absolute refractory period, which is I cannot have another action potential because I'm in the middle of one. And then there's a relative refractory period where if you have a really strong stimulus, you actually can send another action potential. Let me show you what I mean. When the action potential is happening, this absolute refractory, you cannot send another signal right now. No amount of pain is great enough that you could actually keep sending. That's it. However, and normally you wouldn't send one during relative refractory, but let's say there's a lot of pain or a lot of weightlifting, okay? We're gonna send a strong signal. We actually can send another action potential right here, okay? That would during the relative refractory if it's a really big stimulus, but usually it doesn't happen because to get to threshold, you're going to have a really strong stimulus, okay? Because you're down below -70 millivolts down here, your cell membrane. Okay, so that's absolute refractory is like you're in the middle of a action potential. So as long as the sodium gates are open, nope, not going to happen. Okay, so sodium gates are open, and actually the sodium gates are, even though they're closed, there's an inactivation gate, which we didn't talk about, but we'll just say that all the way until we're down here. That's absolute refractory. Relative refractory only can work when we have a really strong stimulus, because we are hyperpolarized, because we are down below -70. Okay. So this is for any area, any part of the membrane. Okay, so how do we send the signal on, how do we send an action potential? Well, it's kind of like the wave, it kind of travels down the axon. So imagine that this is in your brain. So you've got your, imagine this is your multipolar neuron. This is actually a sensory neuron, but it's okay. Let's just imagine you have this multipolar neuron, looks like a star, and it sends the action potential down the axon, down to your soleus muscle, okay? It's gonna, what you're gonna do is, it's kind of like the wave, you're gonna have an action potential right here, and then the membrane right next to it will have an action potential, and then the membrane right next to it will have an action potential, and then a membrane right next to it will have an action potential, and so on and so on and so on. And all of the action potentials behind it will, sort of like the wave, will stop, right? After that action potential, it'll go back to rest. And that's how they travel. And this is called continuous conduction. This is how it can, that's how the travel occurs. So it travels down the axon, okay? That's all, that's exactly how I would explain it. Now, the question is, who does this? Do all neurons do this? And it turns out, no, unmyelinated axons do it this way. So usually if the axon's short, you'll do it this way, but it's not very efficient. It travels pretty slow. So continuous conduction is slow. The action potential travels slowly. Okay, well, let's compare that with the other way. There's another way to do it. There's not, this way is much faster. This is called saltatory conduction. All right, and you will see it on myelinated. If you have myelin on you, you will see this. So here is, up top, I'm going to send the signal down to my soleus muscle. And this is a true story. All of your muscles out here, all of your skeletal muscles have myelinated axons, okay? So long axons are always going to be myelinated. Okay, so here's a Schwann cell. Here's the axon right here. And here's your Schwann cell. So this is all myelin. Here's a Schwann cell. Okay, so those are all wrapping. Cool. What's going to happen is, and I like to think about it not as jumping, but I like to think about the action potentials as sliding. So what will happen is, you're going to have, here's the axon hillock. Here's the very beginning, okay? You're going to have an action potential right here, right? You'll have an action potential right here. It's not myelinated right here. We have an action potential. So a whole bunch of, I've decided I want to do this differently. A whole bunch of sodium is going to rush in, okay? And we're going to get to +30. We're going to have an action potential right here. And then right here next to it. And then right here next to it. Now, check this out. I let a whole bunch of, I just had an action potential, so I let a whole bunch of positives in, a bunch of sodium. What's going to happen is, because I'm insulated right here, I won't lose any of that sodium. There's enough sodium that will diffuse. Let me do a different color. It's going to diffuse underneath here, slide down underneath. That's why I like to call it sliding. Sliding down to get this part, this piece of the membrane, that this internode right here, this part that doesn't have the next part, that doesn't have a Schwann cell, it will be at threshold. Isn't that cool? And so then I'll have an action potential. So let's do it. We'll have an action potential here. So sodium's going to rush in. Sodium's going to rush in, get to +30. And then it's again going to slide. There's a bunch of little pluses that are going to slide down over here to this next internode and get me to threshold. And then I'm going to have a whole bunch of sodium that rushes in, again, slides underneath all the way down the axon. It's super fast. So this sliding under is going to make it, or skipping from one node to the next node, to the next node, to the next node, is going to make it much faster. So this is way faster, but only myelinated axons can do it. Cool. This actually isn't in our notes. So we're on page 3 of our notes, but this is not actually, we won't be tested on this, but larger fibers tend to have really fast speeds. Larger fibers tend to have super fast speeds, but we're not going to worry about that. All right. Okay, synapses. So you can see there's a comparison of muscle and neurophysiology. It's just nice to see that nervous cells have a faster conduction speed. They have many different types of neurotransmitters. So it's just nice to kind of look at this. The resting potential of nervous tissue inside, the resting neurons have -70. It turns out that muscles have -90. So anyway, just it's nice to be able to compare that. All right. Then the last part on page 3 is just talking about neurons and sort of just cleaning up anything that we missed. So let's say I have a presynaptic neuron. Let's say I've got 2 neurons talking to each other. And I'm actually going to draw it out. I mean, I see what they're drawing out there. I see these pictures, but I don't really like them. So I've got a neuron here. Okay, so I have a neuron here. Those are little dendrites. And this is the nucleus. And here's an axon. And here's an axon terminal. Okay. And here's another neuron. Okay, and those are the little dendrites. And there's a nucleus. And there's the axon. And there's the axon terminal of this neuron. Okay, and it's connecting to other neurons down below. Okay, there's the third neuron, and it's axon, and here's its nucleus. And there's neurons here. You know, there's a bunch of neurons everywhere, right? Okay. So if I'm looking at these 2 right here, this is the presynaptic neuron right here. This is the presynaptic neuron. And this is the postsynaptic neuron. So it just means, you know, here's 1 and here's 2, okay? And so the presynaptic, so we're just going to kind of talk about this. And this postsynaptic neuron could be a muscle, right? We're just doing a neuron right now. All right. So it's anything, we're synapsing with anything. So I'm going to send the action potential where my cell body is going to have a graded potential. It's going to get the axon hillock to threshold. Then let's just say I'm not myelinated. I'm going to send this action potential with continuous conduction down the axon. When I get to the end, that action potential, it's going to reach my little axon terminal. Now, what happens at the axon terminal is a part we could clean up. So the axon terminal, when the action potential comes, we have calcium voltage gated channels. That is number 3. Number 3 says depolarization. That means that the action potential is coming. And so I'm going to reach, it's sort of like a threshold, but anyway, I'm going to be depolarized. This action potential is coming. The voltage is different. Okay. I have voltage gated calcium channels on right here on my little axon terminal right here. Okay. I'm going to do a different arrow in blue. I have voltage gated calcium channels right down here. Those voltage gated channels are going to allow calcium to move. The calcium, that calcium moving in to the little, let me change the word on that, to the axon terminal is going to cause the axon terminal to release the vesicles, exocytosis. It will release the neurotransmitter into the cleft. So I will have neurotransmitter released into the cleft, okay? And then it'll go on to the dendrites of the next cell body. Now this cell body will have a graded potential. If it's big enough, it will cause an action potential to happen, threshold to happen, action potential to happen along this axon. Everybody see it? This is a great project idea. Another project idea to put all of this together, okay? The neuron receiving, I'm not that worried about calling it input and trigger and conduction zone, actually. So I'm not that worried about the top of page 4 about those names. I'd rather use axon hillock and then the axon is where the action potential happens, and then the axon terminal is where I release the neurotransmitter. But I would like you to know about calcium and about calcium voltage gated channels, okay? Cool. Just remembering that, just looking at it one more time, that this is your axon terminal, this is a synapse, this is the synaptic cleft. These are the neurotransmitters. We're about to learn about a bunch of them. Good. All right. So neural integration. What is neural integration? This is happening on the cell body and the dendrites, okay? So this is happening, this is when we're talking about graded potentials, okay? So here's my cell body of my neuron, okay? And there's the little nucleus, and these are all dendrites. This is a dendrite, okay? And I'm receiving, I've got a neuron over here. I have a axon terminal of a neuron over here. There's another neuron giving me stuff over here. So this little neuron might have 10,000, might synapse with 10,000 other neurons. I'm not even kidding, okay? And they're all gonna dump, I'm gonna put neurotransmitter in red. They're all gonna be dumping neurotransmitter, possibly, they can, depending on if you're thinking, if you're feeling, if you have sensations. So they might be dumping neurotransmitter on this little cell body and on the dendrites, okay? When I add up all of that, I'm doing something called summation. I'm summing how much of this neurotransmitter, I'm summing how close it's getting me to threshold or how further it's getting me to threshold, okay? So we call, when I'm getting more excited and I'm getting closer to threshold, who's we? The dendrites in the cell body. When they're getting closer to threshold, we call it an EPSP, an excitatory postsynaptic potential, okay? So if I add up all of the pebbles, all of the neurotransmitters and their effects on me being the cell body, and it's excitatory. If I'm normally -70, but because I'm getting excited, I'm -63 on the inside, okay, millivolts. I'm closer to threshold, so this is called an EPSP. However, there's also neurotransmitters that we haven't seen yet, but there's also neurotransmitters that cause me to be quiet. So if I start off at -70 at rest, but I add up all of these neurotransmitters and I'm now -85, do you see how that's inhibitory? I am way more inhibited. I'm not going to send an action potential. In fact, it's much harder. So this is called an inhibitory postsynaptic potential, okay? So EPSP, and you can look at this graph if you want. I don't actually find it useful, but EPSP and IPSP, where do they happen? They happen on the cell body and dendrites, and what is their function? Temporal summation is just if I was to add this all up over time. Spatial summation I find much more interesting, which is over the whole cell body and the dendrites, I add it all up to get to my number at the axon hillock, okay? Good. All right. So summation. We're going to add up all the potentials, and it occurs at the trigger zone. Now that occurs at the axon hillock, so you can use the same thing, axon hillock. So temporal summation means that over time, you're getting more and more neurotransmitter. Spatial summation means that there's a whole bunch of different neurons sending stimulus to you. What I'd like you to be able to do is see it kind of visually or be able to describe it, that I have 10,000 synapses and they're all sending me neurotransmitter, right? They're all sending me neurotransmitter. And I'm going to add up everything that happens, the pluses and minuses, everything that happens to my potential. Am I getting further from, I'm at -70, am I getting further from threshold or closer to threshold? And that's going to all add up at the axon hillock to determine if I'm going to have all or nothing action potential. Okay. Okay, now neurotransmitters. Neurotransmitters. We're going to talk about neurotransmitters. But don't forget, a neurotransmitter is going to have its impact on a neuron. So here's the axon terminal of one neuron, and here's another neuron. This is the postsynaptic neuron. There's the nucleus, and here's its axon. Okay, so I'm going to dump neurotransmitter in purple. I'm dumping neurotransmitter. Okay, now there's 3 ways to remove the neurotransmitter. So neurotransmitters don't stay in the cleft very long. They don't stay in the synapse very long. One way is if it's a really small neurotransmitter, it'll just diffuse out, and other cells like astrocytes will pick it up, okay, and it'll die. So that's one way to remove it, okay? There's another way to remove, which is enzymes. So acetylcholine, our enzyme that, or excuse me, acetylcholine, our neurotransmitter that we know about from muscle contraction. Let's draw it here. Let's draw it like yellow. Okay, so here it is. There's an enzyme that comes called acetylcholine esterase, ACHE, acetylcholine esterase, and it comes and it breaks it apart. It breaks them apart so that they break down. That's another way to get rid of it. That's another way to get rid of your neurotransmitter, because they don't stay in very long in the cleft, like milliseconds. Another cool thing that you can have is on your axon terminal, think about them kind of like they have little vacuums. On your axon terminal, you can have, we're going to release, here's our neurotransmitter in red. You can have little vacuums. They're called uptake transporters, and they uptake the neurotransmitter back. They're like, no, wait a second, I'm going to suck it back in. And they suck it back in into little vesicles again, back in, so they can use it again, okay? So those are the 3 ways that we can clear the cleft. Those are the 3 ways that we can clear the neurotransmitter out of the cleft, okay? Cool. All right, let's talk about different neurotransmitters. You've probably heard about a lot of them. You don't need to ever draw them. But what you want to do is, if you look at page 5 in your notes, you want to have some idea of the ones that we go over right now, okay? So acetylcholine, you already know about. It's it's not always excitatory, but you know about it being excitatory, and you know acetylcholine. It's usually excitatory. It's excitatory on skeletal muscle, but you're going to see it's also inhibitory on cardiac muscle. So it depends on where it is. Another thing about acetylcholine is it's broken down by ACHE, an enzyme. Okay, it's cleared by an enzyme. All right. Others, glutamate. Glutamate is a neurotransmitter. It's tiny. It's excitatory. And that's kind of all you need to know, is it's excitatory. Aspartate is also excitatory, and it's small. These are little amino acids. It's small. GABA is the big one, is the big inhibitory. So it's very inhibitory. And it's an inhibitory neurotransmitter. What does that mean? It gets me further away from threshold. You release a lot of GABA when you go to sleep. Got to get your neurons not firing, not sending action potentials. Very common. Valium uses, so when people take Valium, if you've ever heard of Valium, prescription drug, it enhances the GABA's action. Diazepam is the name of it. Okay, thanks. Okay, now next one. Next group, epinephrine and norepinephrine. They're very similar. If you've ever heard of adrenaline, that's when they're being released as a hormone. So right now we're talking about neurotransmitters. From a neuron is a neurotransmitter. Hormones are released by endocrine glands. So you'll talk about epinephrine as a hormone later. But right now, as a neurotransmitter, it's epinephrine, norepinephrine are excitatory. You can group them together. Dopamine, you may have heard of before. It's kind of the reward, pleasure, addiction. And by the way, anybody can get addicted to anything. Like I have an older neighbor that coupons is a couponer. And I'm pretty sure she's addicted to couponing. I'm not even kidding. So you can be, it doesn't matter what you're addicted to. Some people are addicted to video games. Other people could care less about video games. But the dopamine in your brain is the same if you like couponing or you're a gambler or whatever. If you have that addictive experience, I sometimes have it with sugar. That dopamine is that addiction and pleasure. Histamine. Histamine is another one. It's histamine is definitely excitatory. Probably won't talk that much about it. ATP is excitatory. Serotonin is another big one you may have heard of before. Serotonin, where is serotonin? Oh, sleep induction, appetite, mood control. This is, if you've ever heard of ecstasy, people take them at raves. Also, serotonin is, so ecstasy causes more serotonin to be released. There's also antidepressants like Zoloft, Prozac that work on serotonin and having it sit in the synapse, sit in the cleft longer. So your experience of feeling okay, sleep induction, appetite, mood control, kind of that feeling of somewhat satisfied feeling, okay? A little different than dopamine. Dopamine's more of the addiction feeling. Good. Okay. Last group is the endorphins. So these are big. These neuropeptides are big. And endorphins you've probably heard of. That's the only one that I think you need to know. I didn't go over nitric oxide, so I'll skip that one on the bottom of page 6. Top of page 7, endorphins are part of the opioids. And yes, you may have heard of the opioids, morphine, oxycontin, oxycodone. What other opiates are there? So there's prescription opiates, there's illicit drugs like morphine, heroin. Sorry, morphine's what we take is the prescription. Heroin is what we take, is illicit. So all of these drugs are opiates. They're all working, they're all the same group, and they're all working on your mimicking the neurotransmitters of endorphins. And what is that doing? It sort of has the painkillers, pleasure, painkiller and pleasure center. So if you want to release endorphins naturally, you can do, if you do like a sport where you do it for a while, like let's say you run around for like marathon runners, they often have an endorphin rush. They have that feeling, that high feeling naturally. Yeah. But these are very addictive. As you can imagine, these are very addictive. Yeah, because the feeling is pretty great. Okay. All right, so how do they work, right? So they're moving across the cleft, they go to the other side, they bind to the other side. So I've got a few here that I'd like you to know about in terms of drugs that are out there, and it's kind of fun. So one way we can modify, so pharmaceutical companies and drugs in general, okay, illicit drugs in general, they often modify or change the way a neurotransmitter works. So one way you can do is block or enhance what happens at the synapse, right? So here I am, here's my neuron, my dendrites, and here's the axon terminal of another dendrite, of another axon, right? Here's the axon terminal of another neuron, okay? And they're dumping some neurotransmitter. Let's say they're dumping, DA stands for dopamine. Let's say they're dumping dopamine, okay? Just a little bit. Methamphetamines, if you've ever heard of methamphetamines, that's an illicit drug, causes more dopamine to be released. What kind of an experience would you have if you have more dopamine, right? That sort of pleasure and addiction. And also more norepinephrine, so they're more of awake, arousal, excitatory. So as you can imagine, a brain that's on methamphetamines is going to have that experience. The problem is when we get off methamphetamines, these neurons that we're releasing a whole bunch, they get tired and they release less. So people tend to feel really depressed after methamphetamines. Okay, cocaine. So you can also stimulate or inhibit removal from the synapse. So cocaine, the way cocaine works, it's actually different than meth. So cocaine, let's say this is the normal amount that's released. What cocaine does is it makes it so that there's a bubble here and that doesn't get removed from the cleft. So we just have the experience longer. But these neurons don't get tired because they're not releasing more. So that's why you tend to see the withdrawal symptoms, a little bit less withdrawal symptoms from an individual on cocaine, because those neurons aren't tired. Not to say that cocaine is, I'm not advocating cocaine, I'm just saying it is a slightly different, it's different. If you've heard of MAOIs, or Prozac uses MAOIs. So MAOI is a monoamine oxidase inhibitor. And so what they do is they inhibit, serotonin is the big one, but they inhibit the group of monoamines, mostly serotonin, from being removed. So what will happen is, let's say this is your normal amount of serotonin. Normally it's removed pretty quickly, but Prozac inhibits it from being removed. And so you'll have serotonin sitting in the cleft longer. What is that going to do? Mood's going to feel better, more appetite. Oh shoot, I just removed the whole drawing. You know, I love to draw. With SSRIs, selective serotonin reuptake inhibitors like Zoloft, what'll happen is you've got remember those little vacuums? It'll close the vacuums. So here's my vacuums on the axon terminal. It closes down the vacuum. So it allows the serotonin to sit in the cleft longer. So it's a different way of allowing the serotonin to sit and not get sucked out. That's how there's more than Zoloft, these selective serotonin reuptake inhibitors. These two groups are very common for antidepressants. Okay, one last type, and I put a lot more in your notes. These are the only ones that you need. Okay, it's the notes. The rest of it is just reference about all these different types of drugs, because it's so interesting. This is just all we have time for. So mimics, I thought it was interesting. Isopropanol, sorry that I spelt it wrong. I can see I spelt it, but inhalers, okay? So if you know somebody that has asthma and they have an inhaler, what they're dealing with is a mimic, a neurotransmitter mimic. And so what happens is your airways, when you release epinephrine, norepinephrine in your airways, they dilate naturally. This is you naturally, okay? So you and I have, if we don't have asthma, we generally have nice big airways. What'll happen is often in an asthmatic, those airways start to constrict for many reasons, but they start to constrict, okay? So less air, right? We start to panic. What that inhaler does is it dumps neurotransmitter onto these junctions, these smooth muscle junctions, so that, and mimics norepinephrine. And guess what that does? Causes it to dilate. Yeah, so it's a mimic. And so, and same with what we said was morphine is a mimic of endorphins. So we have a lot of these drugs that we create, prescription drugs that we create that mimic the natural neurotransmitters too. So all the rest of them are just fun. You can look. Parkinson's disease, we have all kinds of ways now that drugs alter neurotransmitters. And that is it for this chapter. I hope you loved it. Lots of stuff here. We'll talk about it more in class.